All right, so I know we are all hungry, and I know we're hungry for web-based visualization and analysis tools, but we're also hungry for food, so I'm going to, see if I can get through these slides pretty quickly. But you are welcome to follow along. Here, you can just go to github.com/Magland or use this QR code if you want to follow along in your. So if I go through the slides quickly, you're welcome to linger on some slides or click on the links. The idea is to have access to this data, so there are links to the different plots. Okay, so why use web-based software? I think we all understand that it's typically easy to use because there's no installation. You can copy-paste links to share things with other people. It's cross-platform. There's advantages for development, and it integrates very nicely with tools like Dandy. There's limitations as well, but I'm not going to focus on those at this point. So I'm going to introduce 3 tools I've been developing for neurophysiology visualization and analysis. The first is FigURL, which is a framework for creating shareable interactive visualizations. The next is NeuroSift, and that's for browsing NWB files, especially those that are hosted on Dandy. And then ProtoCast is a prototype web app. So that's why it starts with the word proto, for analyzing the neurophysiology data in the cloud or with local or cluster compute. So I'm excited to get to that third one because that's sort of what I'm working on right now together with Catalyst Neuro. So an overview of FigURL. Basically, it simplifies sharing of interactive figures. You run a Python script. The Python script generates a URL, and then you can share that with somebody else. And you can create custom visualization plugins for this. So it's supposed to be modular, and it promotes scientific reproducibility and collaboration. So right now I've focused on neurophysiology, but this framework could be used for other domains. So here's a quick example. If you create a plot in Plotly or Altair, a Python library, you can wrap that Plotly figure in a FigURL object and then generate a URL from that, print the URL, and then you could, if you were following along on the presentation, you can click that link and you get the Plotly figure. So it can't really be simpler than that, right? You just get a link and you share it. Of course, the data has to go somewhere. And so the way that works is when you're generating the figure from Python, the data goes into the cloud into a content addressable storage database called Cachery. And then the link points to the browser, and the browser pairs that piece of data in the cloud to a visualization plugin. So really, it's a pairing between a piece of data that's needed for a visualization and a visualization bundle. So fortunately, Alessio Bacino helped with this Spike interface integration. So if you're using Spike interface to interact with recordings or sorting data for electrophysiology, you can either export as matplotlib, IPy widgets, or the third option is through sorting view or FigURL, and that will, again, generate the URL for you to share. So the way that Spike interface integration works is that you define your widget, and then you execute widget.url, give it a label, and then you can print the URL to share. So here's an example of a sorting view / Spike interface visualization that was generated for FigURL. And you can see that there's a lot of different synchronized kind of views, the autocorrelograms, the average waveforms, the spike amplitudes, and there's even the ability to perform curation here. So again, if you click on the link here, you get that live view, and you can start to explore the data immediately. So that's what this is all about, is to really make the data accessible through a shareable link. There's other examples on the FigURL gallery, but we're sort of limited by the development of visualization plugins. So far, I've created all the visualization plugins that exist, but we need more developers, contributors. So I'm not sure how that will happen, but the framework is there for the ability to create these plugins. So average waveform, spike amplitudes, as I mentioned, autocorrelograms, unit locations, and cross-correlograms. All these are interactive, so if you click on one unit in one of these views, the same unit highlights in the other views in that way. So a little bit note about cachery. It's sort of like, I would like cachery to not exist, that there would be some just magic that the data goes into the cloud and then it comes down to download. But of course, there's going to have to be some system that manages manages the data. So cachery is a serverless infrastructure. So it's pretty lightweight system where you just request, well, let me just go to the next slide here. So if I want to add a piece of data to cachery, I create the file, and then cachery cloud store the name of the file, and what comes back is the hash to the file, which then can be used to retrieve the data later from a different computer. Or from Python, you can just say store text or store NumPy or something like that. It goes into the cloud. Let's just pretend that it just magically goes into the cloud and is in some bucket there for now. So when you want to retrieve the data, you can do cachery cloud load from the command line, or from Python, you can do cachery cloud load JSON, load text, etc. And this is what is the underlying kind of system behind FigureL. Okay, so moving right along, I'm now going to the next piece of software called Neurosift. So the goal here is to be able to browse the NWB files that are hosted on Dandy and elsewhere, but also to be able to view those files locally, or if you have NWB files locally that you haven't yet uploaded to Dandy, then you can also visualize them there. So the important thing is that this is client-only application, so there's no server that needs to be maintained. So I have a GitHub repo with Neurosift, and I use GitHub Pages to serve the web app, and there's no other infrastructure that's needed because the web app reads the data directly from the cloud buckets through Dandy, and again, there's no intermediate layer that's needed. So in Dandy, there's a Neurosift integration. So if you go to one of these NWB files, you can do it now if you want. You can click on the external services and then click Neurosift. And that will bring you to this type of view here where you can really explore all the contents of the NWB file. And for those pieces of data that are neurodata types that are supported by Neurosift, you can then click on those and you immediately can start to browse the underlying data. And of course, there needs to be visualizations for that data type that you want to visualize, but there's a plugin system for that. It works similar to NWB widgets, except the difference is that it's on the it's in a client-only web application. So the trick here is that we need to lazy load the data from the remote HDF5 file because the browser is not going to be able to download the 500 gigabytes of the NWB file. That just doesn't make sense. So we want to be able to click on the link and very quickly be able to start to browse the file. And to do that, we have to have an efficient method of loading the bits and bytes from the HDF5 file. So there's some technical details about how I accomplish that. But it's basically a fork of the H5 WebAssembly library. So it's just some examples from Neurosift. You can view raster plots of the spike trains. You can have synchronized views. So this is a spatial series where you have like these kind of trace views, but synchronized with a 2D view. So there's also, this is one of the more advanced views. If you select data types that would be able to come together to form a peristimulus time histogram, then you can interact with this widget. Again, it's sort of a higher level widget than just viewing the individual neurodata types. So there's other examples here. So this is sort of a list of all of the neurodata types that are currently supported. And you can go to the gallery to kind of click through those and see examples of how they work. So viewing local files, we try to make this as easy as possible. So it's pip install Neurosift. You also have to have a modern version of Node.js. And then you can do Neurosift, view NWB, and then the path to the local NWB file. And it will give you the same interface as though you're looking at the file on the remote site. OK, so trying to go quickly through here. Protocast is, as I said, it's a prototype web-based tool for analyzing the neurophysiology data in the cloud or with local compute resources. So other similar projects are Neurocast and Spike Interface Cloud. So it's really under heavy development now jointly with Ben Dichter and Luis Taufer at Catalyst Neuro. But the idea is to enable labs to use cloud resources to run analysis pipelines on their data and encourage them to upload to Dandy in order to make that work. And the initial focus is on spike sorting, but we can also plan to expand this. So the steps are kind of as simple as we can make it, right? So you create a Dandy set. So you can use a system like Guide or other system to import your data into NWB and upload to Dandy. The step 2 is to then import the Dandy set into Protocast. It doesn't create a copy of the data. Protocast will just have links to those files. The step 3 is to launch the spike sorting from the web GUI, configuring a compute resource. So you specify, do you want to have it run in the AWS cloud, or do you want it run on local resources? Then you can browse the outputs with Neurosift and then ultimately upload the outputs to a new Dandy set so you can share or archive what the spike sorting output is. So this is what it looks like to create a project. You could actually give it a try if you're following along. You log in with GitHub, create a project. Then you can click on the Dandy Import tab on the left, which allows you to browse the Dandy archive. So it's great that Dandy is built on a RESTful API that's open, so that allows this web app to be able to use it directly. So you can browse through the Dandy archive, select the NWB files that you want to import into your project, and click Import Selected Assets. You then have this view. Again, these are pointers to the files which reside in Dandy, so the data is not actually copied over. For spike sorting, you then select the NWB files that you want to run spike sorting on. You select the spike sorter, for example, kilosort3, specify the different parameters, and run the spike sorting. You can monitor the process of the jobs that are running. This could be on a Slurm cluster or a local machine. And once the spike sorting is complete, you now get new files that have been added to the project. And the naming convention, and Yarek will be very pleased to see this, that we've appended onto the name of the Dandy asset _DESC description dash, and then the information about which spike sorter was run. So this data now is ready to be imported directly back to Dandy because of that naming scheme. So it really makes it as easy as possible. Now, of course, we don't want to make it too easy that people don't actually look at their data. So that's why the data doesn't go directly to Dandy. You then have the ability to view it, interact with it, and maybe even run it through your pipeline and everything. Once you're satisfied that this output is worthy of sharing on Dandy, then you click Upload to Dandy. You provide your Dandy API key, and then just click Upload, right? So then in the metadata on the Dandy archive is all the provenance of the steps that were used to create that sorting output, so somebody else could reproduce it. I should also mention that within protocasts, you also have a record of all the jobs that were run and their provenance. So I think I'll skip this slide because you can kind of review it, but this is kind of a list of some of the advantages that Ben and I came up with about using this system in terms of scalability and giving people access to these processing tools. So maybe this is the most exciting slide here. So these are some initial experiments with protocasts. And so I've run, maybe you just can't see it that well, but you can see here it's kilosort 2.5, kilosort 3, kilosort 2.5 first 10 minutes, for kilosort 3 first 20 minutes, mountain sort 5, et cetera. And now you can now it's kind of well documented in a place where everybody can access it, right? So we can run a Python script that accesses this data and just asks what is the consistency between these two sorters, and begin to come to a consensus on, how consistent are the different methods, which units are reliable, et cetera. So I'm really excited for the next steps forward in terms of using this tool to answer some of the questions about what spike sorter should we use, because the Dandy archive is a great resource where we have these standard data sets and where we're all looking at the same thing instead of each lab saying, okay, I ran it on my data and here's the results. Instead, we can have like a central place where we're running the spike sorters, everybody can see what was run, and we can begin to sort through what's happening. So there's also some exciting things about how to create protocast processors and how to allocate your own compute resources. But in the interest of time, I would just like to thank my collaborators, and thanks for your attention.